Okay, good day, everyone. Welcome to our new MNS webinar. Today marks a special moment for our community as we celebrate the inauguration of the Glia and More Resource Group for MNS members. This group is created as a focused space for discussion, collaboration, and future scientific activities centered on Glia, glial biology, and related fields. Within this context, we are very pleased and honored to host a webinar by Professor Alexei Verkhratsky, who will speak on the physiology of astroglia. His work has shaped much of our current understanding on glial cells, and we are very delighted to have you with us, Professor Alex. Before the webinar, we are happy to introduce Professor Mariagrazia and Professor Valerio Magnaghi, who will give us a brief account or a brief overview of the expected activities of the Glia and More Resource Group, and then introduce Professor Alex. The webinar will last for 40 or 45 minutes, and then it will be followed by a live session of Q&A. And so we welcome everyone here, and we encourage you to participate actively and share your questions at the end of this talk. So without further ado, we welcome together Professor Valerio. How are you?
Okay, thank you, Nermeen, and also from my side, good afternoon and welcome to everyone attending this webinar and this celebration. And first, I wish to thank the actor and the past presidents of the Mediterranean Neuroscience Society that enthusiastically give us this opportunity and supported this our idea to gather all neuroscientists belonging to MNS around the topic of glia and non-neuronal cells. This is our logo, as you can see. Maybe we have to change a bit this logo as suggested, but for today, this is the opening and the first occasion to present our initiative, meeting all people potentially interested in glia and neuronal cells. And actually, our initiative is ambitious and hope to attract much more people than expected, even in MNS. But a prerequisite is to be an official member of the MNS Society, and so we can enter here in the member. This is mandatory, and this is the main web page of MNS, from which you can get into the Glia and More page. And this is the specific page of MNS Group of Glia and More. And you can find the presentation of our group. And basically, our idea was carried out during the last MNS conference in June in Crete this year. And still it actually took shape from our former idea, the idea by Mariagrazia and me, focusing the attention of neuroscientists, not only on neurons, but also other cells, glial cells and non-neuronal cells, taking into consideration the wider vision of this term. The intention is to create a network of so-called gliologists within the Mediterranean Neuroscience Society community. And basically, our main goal is to engage young and senior scientists at every stage of their career on the topic of glia and neuronal cells in the central as well as in the peripheral nervous system, even with the aim to promote discussion and establish new collaboration in the field. And this is the list of the actual group members, including the core of the founders, with the hoping to rise soon with other members of the MNS that are welcome with the topic that are welcome and that could be interested on the topic of neuronal cells in the nervous system. In the next coming month, on the basis of the new members, the next step will be certainly to organize a discussion forum on Meet or something similar together, and moreover to exchange ideas and how to implement the activity of our group. Of course, other webinars will come soon. But today, what I want to do is to launch officially, to inaugurate our group. And we thought to organize this webinar with auspices that our webinar will come very, very soon. But I wish to hand the floor over to my coordinator, co-coordinator, Professor Mariagrazia Grilli, and please, the stage is yours.
Thank you, Valerio. Good afternoon, everybody. I think that Valerio already mentioned probably the major idea behind the group. So I prefer not to wait any longer and to introduce Alex. Eventually, at the end of his presentation, of course, Valerio and I are also available for answering questions regarding the group itself. So for me, it's a great and sincere privilege to introduce Alex Verkhratsky, who is Professor of Neurophysiology at the University of Manchester in UK. To me, and to most of the people, Alex is an absolute legend in the field of neurophysiology. I knew you would have that kind of reaction to my statement. Anyway, this is what I think. Alex is a legend in neurophysiology, is known everywhere for revolutionizing our understanding of neuroglia. So for many decades, glial cells have been regarded as cells living in the shadow of neurons. But Alex changed that narrative forever. His groundbreaking work has been fundamental in establishing glial cells as dynamic and active players in brain function.
So,
of course. Please close microphones. Thank you. So, among his transformative discoveries, Alex, for example, just to give you a sense, was one of the first ones to identify neurotransmitter receptors in astrocytes and demonstrate their activation during physiological stimulation. He was one of the few to reveal initially matching patterns of neuronal and glial receptors, so that it could be established the concept that now we give for granted, the neurons and glia engage in active reciprocal communication. He was the one who demonstrated that these receptors connect to calcium-mediated signaling cascades, introducing the concept of glial calcium excitability. So this just to name a few. Alex also pioneered the field of neurogliopathology. For example, he characterized in the last decade that astroglial asthenia, atrophy, and dysfunction are key early events in neurodegenerative and neuropsychiatric disorders. And as a pharmacologist, I would like to highlight that this is probably opening entirely new avenues for potential therapeutic intervention. I could continue listing, all these impressive achievements in terms of paper, chapters, books. I would like to mention that he was the one to wrote the first comprehensive textbook on glia neurobiology, that he serves as a member in many prestigious academies. But I'm too eager to listen to his seminar, so I think I'll stop here, but not before I share one personal comment with him. Alex is not just a world leading scientist, but he's a colleague and a friend whose passion for neuroscience is matched only by his generosity in sharing his tremendous knowledge. So Alex, thank you very much for accepting Valerio and my invitation today. It was really a sign of friendship. Thank you.
I think that I can stop my presentation and move to the presentation of Alex.
Yes.
Yeah.
And now I can share, yeah?
Okay.
No, it still says Valerio is presenting.
I'm still presenting.
Well, that's...
Yeah, okay. That's okay. You can present yours.
I'll try. How? Select Windows. Sorry, it's first time I do this.
Meet.
Slideshow. Allow. Do you see it?
Yes, we can see
Yes,
it
so
now.
it's all there, yeah?
Yeah.
It says you are sharing another application window. What does it mean?
It was working.
It
Your
was
Yeah,
working.
yeah, I
presentation
know,
was-
but it was blinking in my hands. Sorry for that. I mean, okay, hello. You see it, everything is fine because in my hand it's blinking like I don't know what. Thank you very much, my dear, and good afternoon to everybody. And indeed, thank you very much, Mariagrazia. Thank you very much, Valerio. And as Mariagrazia said, we do enjoy many, many years of friendship, and it is obviously quite an honor for me to be the first speaker in this particular group. And to start all this series, which I believe is going to be really exciting because it is a new forum in which we can discuss many, many different things concerning the brain, concerning the structure of the brain, how it works, and the role, of course, of neuroglia, which is my long-lasting passion. So before we start, I would like to remind you that we are at war. And unless this brutal country is fully destroyed, our world is in peril. So Russia delenda est, and in that I would like to obviously remember Kata the Elder. And let me start from the brain. And the brain is something incredibly complex. Evolution somehow put into a very small volume of about 1.5 liters an incredibly complex structure which comprises about 200 billion cells, which are connected with many, many, we don't know how many, but very many, probably tens or even hundreds of trillions of connections. And what is truly amazing is that this incredible complex computing machine, which does all sorts of things, which makes us creative, which makes us humans, does it all at a really meager expenditure of about 300 to 400 calories per day, which ends up at about 12 watts per hour. So it's less than a lamp. And if you compare it with a supercomputer, which has a comparable memory and comparable ability to make calculations, that will occupy many hundreds of cubic meters of space and will require a nuclear power plant in order to supply it with all the energy needed for this particular machine. So when people tell me about artificial intelligence and computers and everything, I sort of laugh about it because all computers in the world would never probably match the creativity and capabilities of the human brain. Now, if you look into the brain as an organ, we can see that that is a sort of interaction of many functional networks. And probably the network number one, which makes the brain, it's what is called connectome. Because all these 100 billion of neurons and another 100 billions of non-neuronal cells on their own do not produce the brain. What produces the brain is when all these cells are connected. They're physically connected, and in the brain, neurons are connected, obviously, with axons, many of which are myelinated. And it's sufficient to say that there are about 180,000 kilometers of myelinated axons inside the human brain. So it is really quite remarkable system. And this system is supported by neuroglia. It's supported by oligodendrocytes, which support axons and myelinate them. It's supported by astrocytes, which support synaptic connections, and by microglia, which shape these synaptic connections and which assist proper myelination in the development. Of course, these many neurons require energy, so they require glucose and they require oxygen, and that is delivered by brain vasculature. And again, on average, human brain contains about 650 kilometers of vessels. And these vessels are supported again by astrocytes, which provide glia limitans perivascularis and which regulate the blood-brain barrier, which of course is made by endothelial cells. The brain is also floating in the fluid. And that is of, again, paramount importance because this fluid, first of all, due to buoyancy, reduces the mass of the brain to about 40 grams instead of 1.5 kilograms. And second, this fluid is protecting the brain against bumping against the skull. And that allows us to do all what we do. We can run, play tennis, drive a car, and still continue to think, and our brain is functional. And this part is, again, very much supported by glial cells. This is ependymoglial cells, which are producing cerebrospinal fluid, and ependymoglia, which are taking care of distribution and redistribution of this cerebrospinal fluid and normal functioning of this particular system. And finally, there is an alternative connectivity system, alternative to the neuronal connectome, and that is panglial syncytia. And those panglial syncytia are covering all the brain. And here you can see a very recent image which was published in bioRxiv, in which you can see the extent of panglial syncytium. It really covers the whole brain. So a single injection somewhere in hypothalamus resulted in spread of fluorescent proteins across most of the brain, as you can see here. And that panglial syncytium is supported by astrocytes and oligodendrocytes. So as you can see, neuroglia is central for functioning of major networks which make the brain and which make the brain function. So the brain contains very many cells, and these cells are making, of course, intricate connections, and we hold that all these cells are equal. So these cells are born together, they develop together, they learn together, and they die together. So in a sense, when we are talking about how to study the brain, we have to understand that we are studying the nervous tissue as a whole. So we have to always think about feedbacks and feed-forwards, which are tailoring and shaping all these cells in one single entity, which produces brain function and which we call as brain active milieu. So a couple of historic words. What is neuroglia? That was born in 1858 when Rudolf Virchow presented it during the lecturing to students of Charité. And he believed in neuroglia as a connective tissue which surrounds, which forms the basic of the brain into which all other elements are embedded. So in a sense, for Virchow, it was not really clear whether it's acellular connective tissue or cellular elements. So he used in his book sometimes the word glial cells, but he also very much emphasized that it acts as a connective tissue. And he was, of course, a proponent of connective tissue and of its physiological and pathophysiological meaning. So what is neuroglia? It's a highly heterogeneous population of neural cells, which are macroglia, astrocytes, and oligodendroglial cells, and ependymoglia, of non-neuronal origin, which are microglial cells. But all these cells are responsible for brain homeostasis, for supporting the connectome and myelination, and all of them are contributing to the defense of the brain in pathological contexts. So that is a classification. Neuroglia is subdivided into a peripheral nervous system glia and central nervous system glia. And the periphery, we have different sort of Schwann cells, satellite glia and pteriglia. In the central nervous system, we have macroglia, astrocytes and oligodendrocytes and glia of special organs, which are of ectodermal neuroepithelial origin. And invaders, microglial cells, which are myeloid cells, which enter the brain very early in development. And today, I will mainly concentrate on astroglial cells. Of course, numbers, this is really very important. And still, you can see in many textbooks and in many papers that in human brains, there are 10 times more glial cells than neurons. That is false statement. The total numbers are more or less the same. Actually, it's a little bit more neurons than neuroglial cells, but of course there is a huge regional differences. And here you can see numbers between mouse and human, and you see in mouse there are millions, about 70-75 million of neurons and about 40 millions of non-neural cells. In human, it's 80 to 100 billion of neurons and maybe 80 to 90 billion of non-neural cells, but we should not forget that these neural cells also cover pericytes and also cover endothelial cells, which are very numerous. There are probably about 5 to 10 billions of these cells. So that's about numbers. Introduction into astroglia, and astroglia stems to Camillo Golgi, who introduced the black stain technique. which allowed to open the brain, which allowed to stain cells and to see their fine structure. And these are how the cells were looked when stained by Golgi technique, and many of them, they had a sort of stellate morphology. And also Golgi realized that many of those cells are sending processes to the blood vessels, and these processes are ending up with end feet, and that led him to a conclusion that these cells, glial cells, future astrocytes, are important as an interface between the blood and the brain parenchyma, which exactly is one of the archetypal functions of astroglial cells. And based on these images, in 1895, Michael von Lenossek introduced the word astrocyte, which literally, of course, means star-like cell. But he had a prophetic mind, because what he wrote in his book, he wrote, I would suggest that all supporting cells be named spongiocytes, and only some of them astrocytes. And that is incredibly important, because indeed, if you do a proper staining and proper visualization of astrocytes, they don't look like stars at all. They look like bushes and sponges. And therefore, please never start your paper with a statement that we are looking at a star-like or star-shaped cells. They are not, despite the fact that the name, of course, is there, and we still continue to call them astrocytes. Astrocytes belong to a larger class of glial cells, which are called astroglia. And those astroglia covers, of course, very many parenchymal astrocytes, which are many different morphotypes. Here you can see only some of them, such as protoplasmic, fibrous, or velate, or perimeningeal. They also include radial glia, which exists only in developing brain of the fetus, and then it disappears after birth. There are very many radial astrocytes, which retain sort of radial morphology, like Bergman glial cells in cerebellum or Muller glial cells in retina. And there are also very important radial glia-like neural stem cells, which, although everybody calls them neural stem cells, they are not their astrocytes with stemness, because besides their capability of producing asymmetric division, they also make all functions of astrocytes. They contact synapses and contact blood vessels and make glial limitants and so on and so forth. So our stem cells in our brain are actually astrocytes. And then there is a whole class of ependymoglia, which formally belongs to astroglia, but I more and more think that we have to make them a separate entity because of their absolutely topical localization. So these cells are making a single cell layered layer of the walls of ventricles and the central canal of spinal cord. And they are subdivided further into ependymocytes, proper ependymocytes, choroid plexus cells, which produce CSF, and tanycytes, which are sitting at the walls of the third ventricle. And finally, high primates and humans possess several classes of astrocytes which are not present in any other species, and these are interlaminar astrocytes, varicose projection astrocytes, and polarized astrocytes. What astrocytes or astroglial cells do, they do take care of homeostasis of the brain at all levels of organization, starting from molecular homeostasis, cellular, so they control synaptogenesis, they control neurogenesis, metabolic homeostasis, organ homeostasis, they control lymphatic flow and blood-brain barrier, and even systemic homeostasis, because very many astrocytes are chemosensors, and they are involved in telling neurons what's going on with milieu interior of the body, and then neurons, of course, do whatever behavioral or energy-dependent tasks which are rectifying these particular things. And finally, astrocytes are central for the defense of the nervous system, for neuroprotection and regeneration. And when astrocytes fail, that has a dire consequences, because neurons are dying and they cannot survive without astrocytes taking care of their homeostatic needs. How to visualize astrocytes? That is not a particularly easy task. Of course, good old Golgi staining remains. Now we have to remember Golgi staining is a sparse staining. It doesn't stain all cells in the brain, but only some of them, maybe 5 to 10%. But in good hands, Golgi staining can produce an extremely good images and can be used even for electron microscopy. If you want to count all the cells in the brain, we have to use Nissl staining or Gimsel staining, and that is of course a problem because you need a very highly qualified histologist to tell what is what. Nonetheless, this is so far probably the only really accurate stereotaxic technique for counting all the cells in the nervous tissue. Immunocytochemistry is of course of big help. And somehow it's also universally believed that glial fibrillary acidic protein, or GFAP, is universal marker of astrocytes. This is not the case. GFAP in normal conditions, antibodies are staining probably about not more than 20% of all astrocytes. It doesn't mean that they don't express GFAP, they do, but they express it below the level of detection. And secondly, GFAP doesn't reveal morphology of astrocytes. It works very much like Golgi staining because it stains interlaminar filaments, intermediate filaments, sorry, which are a part of cytoskeleton. So with GFAP, more or less, we visualize the cytoskeleton of those cells. So other types of antibodies are needed, and there are different markers such as protein S100, for example, or glutamine synthetase, which is highly astrocyte specific, or enzyme, which is also astrocyte specific and stains only peripheral or mostly peripheral processes, but none of them really reveals the full morphology of astrocyte. To reveal the full morphology of astrocyte, we have to either perfuse this cell with fluorescent dye, such as lucifer yellow, for example, or we have to express fluorescent reporter under control of astrocyte-specific promoters, which are several. And in this situation, we see this image, which is at the bottom of this panel, and which really shows how astrocyte looks like. It looks like a big, big, big sponge. And that is because astrocytes has, many astrocytes, protoplasmic astrocytes, they have a tiny, tiny peripheral processes which are below the level of detection, but still, of course, they emit fluorescence. And that what makes astrocyte, because with the help of those processes, astrocytes are sensing synapses, they cover synapses, and they also establish connections between astrocytes in the form of panglial syncytia. Astrocytes are highly diverse, and that was clear already from the very first morphological studies of those cells. And here you can see Golgi staining of human fetal brain, which was made by Gustav Magnus Retzius, who was in Stockholm of those days. And you see incredible diversity of morphotype of astrocytes with different soma, with different arborization, with small processes, large processes, and so on and so forth. So astrocytes are highly, highly morphologically different. And there are many, of course, major morphotypes of these astrocytes, such as I already mentioned, radial glia. And these radial glial cells could be very different. So as you can see here, Bergman glial or Fanana cells in cerebellum, which are feathered glia, or also in brainstem we have radial glia, and in supraoptic nuclei, and in neurogenic niches. And you see again how very different they all are. The main type of astrocytes in the cortex, in the gray matter, is of course protoplasmic astrocytes, and those protoplasmic astrocytes, they have a very specific morphology. So they have a soma with primary processes, and then many different secondary processes in larger order and magnitude, which at the end emit these extremely, extremely small processes, which are called leaflets. And those processes are very peculiar because A, they have an incredibly big surface to volume ratio. So their thickness is about 100 nanometers. So it's a very huge surface area with an extremely tiny intra-process volume. And they don't have organelles because they are too small to have organelles. And those are processes which are contacting synapses. And they contact them in all possible ways. So a single synapse can be contacted by one or many leaflets from the same cell or from different cells. So that is all sort of combinatorics. And then protoplasmic astrocytes send at least one or usually 2 to 4 processes to the brain, or to the blood vessels, when they form and fit and make, obviously, the interface with the blood. In the rodent brain, astrocytes have a very peculiar site architecture because each of them occupies an individual domain. So the interlap between these individual domains is very small, not more than a couple of %. And so within this individual territorial domain, everything is covered by the single astrocyte. And of course, every single astrocyte sends processes to capillaries, and then so somehow makes a relatively independent unit, which is known as neurovascular glial unit. Now, that is rodents. It may not be true for all other species. For example, in ferrets, there are no territorial domains. And it is a big question whether they exist in humans. And this is a wonderful morphological structural work in which it was made a total electron microscopy and 3D reconstruction of 1 cubic centimeter of a human cortex. And here you can see astrocytes. This paper was published recently in Science, you can find it. But you can see these astrocytes. There are about 8 astrocytes on the single image, and you see how intermingled they are. So there is no indication whatsoever for individual territorial domains. And that is very important, and we have to look into it, because if that is the case, then human brain is completely different from mouse brain, and all our ideas and concepts of individual neurovascular units, which we derive from brains of mice and rats, may be completely irrelevant for the human. Another issue, of course, the human-specific astrocytes, and here you can see what are the difference. So these are protoplasmic astrocytes, and protoplasmic astrocytes in human brain are about 10 to 20 times larger and many times more complex than rodent astrocytes. And that translates in the fact that single human astrocyte covers about 2 million of synapses, whereas single rodent astrocyte covers about 20 to 100 1,000 synapses. And of course, human brain has a specific astrocytes, and in particular, these are interlaminar astrocytes. Why they're called so? Because they send very long processes. Their somata are sitting just under the pia, and their long processes penetrate through several layers or lamina of the cortex and go down to layer 4 or even sometimes layer 5. And these cells are unknown. We have no idea what they do, but we know that, for example, in Alzheimer's disease, when it comes to dementia, these cells are disappearing completely. So there are no interlaminar astrocytes in Alzheimer's brain. And how to obviously study them in rodents is a big question. One of the possibilities would be to take stem cells, derive astroglial precursors from them, and inject into the mouse brain, which acts as a biological incubator. And in this situation, it's possible to have some mice with interlaminar astrocytes and study them. Physiology, membrane physiology. Obviously, astrocytes are very similar to all other cells in a sense that they maintain ionic gradients across the membrane. So their intracellular ionic concentration is very different from extracellular ionic concentration. In general, this concentration of ions are similar between neurons and astrocytes, with 2 exceptions. Exception number 1, sodium. Astrocytes have about 2 times more sodium than neurons. And second is chloride, and astrocytes may have even 10 times more chloride than neurons. And that is, of course, physiologically incredibly relevant, because when neurons are facing, for example, GABA as inhibitory neurotransmitter, GABA hyperpolarizes the neurons and triggers chloride influx, whereas it depolarizes astrocytes and triggers chloride efflux. And that is important for inhibitory transmission. At the very core of membrane physiology of astrocytes lie potassium channels. Astrocytes express a very high density of potassium channels, so more or less their membrane acts as a potassium electrode. So you see this is a linear IV curve. So whenever you depolarize the cell, whatever you do, that will trigger a huge potassium current, which will very rapidly bring bring membrane potential back to resting membrane potential, which for astrocytes generally is about -80 millivolts, with some exceptions in different brain regions. The second very important feature of astrocytic physiology is that they are connected. They are connected in panglial syncytium, which connects astrocytes, oligodendrocytes, and ependymal. And these syncytia also have some anatomical organization. For example, in the somatosensory cortex, in the barrels, astrocytes within the barrel are connected, but they are not connected to an adjacent barrel. So it's another anatomical structure of the brain which is based around astroglial syncytia. And these syncytia are very important because on one hand, they confirm what is called isopotentiality of the syncytium. So they shunt whatever possibilities, whatever trials to depolarize astrocyte because of a huge diffusional connectivity between different cells. And second, they allow the intercellular exchange of molecules, intercellular exchange of energy substrate like glucose, for example, or second messengers such as inositol 3-phosphate, which of course underlies calcium propagating waves. And here, as I already said, you can see the image of panglial syncytium, and then you can see, so what was that? It was injection with a virus and transfection of astrocytes with a specific protein, which can diffuse through the syncytium, and you see that in response to injection in whatever site, a huge part of the brain is covered, and even an adjacent hemisphere, so it is not staying in one hemisphere but goes to another. And that, of course, adds a huge degree of plasticity and complexity to a brain signaling systems. Ion channels, as I already said, potassium channels are the most important, and astrocytes express several classes of potassium channels, which cover all physiological range of potentials from -80 to +120. At whatever potential, another class of potassium channels will be opened and provide for a massive potassium current to maintain very highly hyperpolarized potential of astroglial cells. A peculiar channel which is very much present in astrocytes is a so-called inward rectifier potassium channel 4.1. And what is inward rectification? It means that the current from the area of high potassium concentration to low is smaller than in opposite direction. What does it mean? That these channels are open and active below resting membrane potential, and in response to depolarization, they close. And the mechanistic explanation is spermine, which really literally blocks the pore of this channel upon depolarization. And those channels are more or less setting up the resting potential of astrocytes. Sodium channels, and that is quite interesting because on the level of expression of protein and the level of expression of mRNA, there are certain sodium channel subunits in astrocytes, mostly in cultured astrocytes, but also sometimes they could be found in slices, and in particular it's NAV 1.5. But so far there are no credible evidence for any voltage-gated sodium channels in vivo in astrocytes in physiological conditions. Instead, some astrocytes, especially in subfornical organs, express a very specific sodium channel, which is called NAX, and this channel is gated by extracellular sodium concentration. And in subfornical organ, as you certainly know, capillaries are fenestrated, so astrocytes can have a direct access to blood, and with the help of this channel, they monitor sodium concentration in the blood. And when the sodium concentration goes up, that triggers sodium current in astrocytes, sodium entry, and this sodium entry somehow translates in more lactate, and that lactate signals to adjacent neurons, which produce whatever behavioral responses. Calcium channels. Again, at the beginning in cultures, there were a number of experiments which identified very small but still voltage-gated calcium channels in cells which were considered to be astrocytes. And they were even found in acute brain slices, as you can see here. But then it turned out that those so-called complex astrocytes were not astrocytes at all, but they were oligodendroglial precursor cells. And those oligodendroglial precursor cells are very different from all other glial cells because they're electrically excitable, and they do have voltage-gated calcium channels. So far, again, we don't have any evidence that voltage-gated calcium channels exist or operational in astrocytes in vivo. What really is operational in most of the astrocytes are calcium store-dependent calcium channels or crack channels, which are known also to belong to Orae family. Orae comes from a name of Ora, which were goddesses which kept the gates of Olympus, and that's how Verkhratsky calls them when he discovered those particular channels. So these channels are governed by changes in calcium concentration inside the endoplasmic reticulum. And for glial cells, endoplasmic reticulum is one of the major sources for calcium signaling. And when there is a release of calcium from endoplasmic reticulum, calcium in it goes down, and that triggers oligomerization of another protein, which is called STIM, and multimer of STIM opens store-operated calcium channels, either Orae or of TRP family. And those are present in astrocytes, and there is Murray Prakriya, a very nice study which shows that these channels are sitting in peripheral processes of astrocytes, and they do maintain spontaneous and evoked activity, which may trigger local calcium signals. Finally, astrocytes have calcium release channels. Mostly it's inositol-3 phosphate receptor channel type 2, which sits in the endoplasmic reticulum and which is responsible for calcium excitability of astrocytes. There's some evidence for ryanodine receptor, but still it's difficult to put it in a context. What is important, however, for those calcium release channels is that they are sensitive to calcium in the cytosol. So they are capable of producing regenerative openings. So they are capable of producing propagating wave of opening of these channels through endoplasmic reticulum. And that is an underlying mechanism for propagating calcium waves. Now, astrocytes, as well as most of other neuroglial cells, with exception of OPC, are electrically non-excitable cells. What it means that they are incapable of producing and generating of action potential. However, they employ a completely different mechanism for their excitability, because these cells are still excitable, but in a very different way. So whenever something comes to the astrocytes, they respond not by changes in membrane potential, but by changes in intracellular concentration of ions and intracellular concentration of second messengers, such as cyclic AMP, for example, or inositol-3 phosphate. And there are many ions which are capable of producing signals. Now, at the beginning was, of course, calcium, and you remember Otto Levy, who in 1959 said calcium is alles, but in astrocytes, calcium is nicht alles, it's not everything, because other ions are producing signaling events and are supporting intracellular signaling and intracellular excitability of those cells. So with calcium, everything began at the end of 1980s after Roger Chen introduced first Queen-1 and then Fura-2, so intracellular calcium indicators, which he did in 1981, I guess. And these were experiments which were performed across the world, and that is one of the very early experiments which was done probably in 1992. And that you can see what we did. We have a co-culture of neurons and astrocytes. We stain them with Fura-2. And then we start to stimulate with different things. And when we stimulated with high concentration of potassium, that obviously depolarizes the cells and opens calcium, voltage-gated calcium channels. And you see what happens here. In response to this stimulation, all neurons become red and white. So it means that they responded it and they generated action potential. But then we did another experiment in which the calcium was removed from the solution and the culture was stimulated by ATP, which is archetypal stimulator or agonist of metabotropic purine receptors, which are producing calcium release from the internal stores. And that's what happened in this situation. Neurons remain idle and all astrocytes become red and white. So that shows the difference between them. So it's in astrocytes, it's intracellular excitability, which at least at those moments was believed to be solely associated with endoplasmic reticulum. And that is the schematics of a classical schematics of classical calcium signaling. In astrocytes, we have endoplasmic reticulum with a very high calcium concentration. And when these cells are facing a neuronal activity, their metabotropic receptors are activated. Inositol-3-phosphate comes down to the endoplasmic reticulum and triggers release of calcium from it. And when this release empties endoplasmic reticulum, it triggers a secondary calcium entry through store-operated calcium channels. So that was the major paradigm. And those signals produced by those agonists were very highly compartmentalized, and there could be a very local or so-called micro or nano domains of calcium signaling. They could be fully global and synchronized, or very important in culture situations, these calcium signals could propagate. So you may, when you have a culture of astrocytes, a confluent culture which covers the whole Petri dish, for example, you can stimulate a single cell in the middle of this culture, and calcium signal will propagate through all the cells in the syncytium. And this propagation is, most importantly, not diffusion of calcium, but diffusion of inositol-3-phosphate or extracellular diffusion of ATP. Calcium cannot diffuse through cytosol because cytosol is extremely rich in calcium buffers. Now, for the time being, the situation is much more complicated because so far we could not really find these propagating calcium waves in the situation in vivo, so it's a big question remaining. It turns also out that mechanisms of calcium signaling in different parts of astrocyte are different. In the soma and in main processes, which are called branches, calcium signaling is mostly associated with calcium release from endoplasmic reticulum and secondary store-operated entry. But in the peripheral leaflets, which, as you remember, do not have endoplasmic reticulum, the mechanism is very different. The mechanism is now plasmalemmal because most of it comes with the calcium which enters through the plasmalemma, either through channels or ionotropic receptors, or most of calcium actually is transported into leaflets through sodium-calcium exchanger working in a reverse mode, which is sodium dependent, as you will see slightly later. When we look into in vivo situation, usually that is something like that when there is some physiological stimulation or locomotion and you look into different cortical area, what happens, calcium goes up and down in a very synchronous manner in many astrocytes within the field of view. And so far, again, as I said, we don't have credible evidence for existence of propagating calcium waves. So what calcium does to astrocytes? Of course, calcium has many generic functions, so it controls and regulates translation, transcription, metabolism, energy production, and so on and so forth. But what it does to astrocytes, and this is a major question, because let's say when it comes to skeletal muscle, it's very simple, no calcium, no contraction. Now, what will happen to astrocytes without calcium? And in early 2000, about 2002 or 3, McVicker and his colleagues made an experiment. They deleted astrocyte-specific IP3 receptor type 2. And that more or less removed calcium signaling from the soma and main branches. And that didn't result in major phenotypes. So there are some changes, but they are not major. So even in the absence of ER-originating calcium signals, astrocytes can perform their function and nothing major happens. So that is more or less a huge question in the field, what really calcium does specifically in astrocytes. Calcium signaling in astrocytes and activity of neurons in certain conditions are linked, but they are very often completely dissociated. And here you can see recordings of calcium in astrocytes and neurons. So astrocytes are on the left and neurons are all on the right. And you can see in astrocytes locomotion, this is in vivo experiment, triggers one massive big calcium signaling, which then disappears and nothing happens. In neurons, locomotion triggers many different signals which are transient, which probably are associated with neuronal bursting, which is triggered by locomotion. So calcium is not everything. And if you start to look into astrocytes, you will see that most of the channels they have are sodium permeable. And even the channels which we tend to call calcium channels, like, for example, NMDA receptors, in real physiological conditions, they produce massive sodium currents, and calcium entry is much smaller. Why is that? Because it's simply way more sodium outside than calcium. It's 100 millimolar versus 2 millimolar. Moreover, astrocytes have an extended complement of transporters and exchangers. And most of these transporters and exchangers utilize the energy of sodium gradient in order to transport things through the membrane. So many different pathways in astrocytes are producing massive sodium entry. And astrocytes have a very specific sodium-calcium pump. The sodium-calcium pump is distinguished from neurons by expression of alpha-2 subunit, and this is purely astrocytic. It doesn't exist in neurons at all. And this subunit is very peculiar. Its Kd for potassium is in a range of 3.5 millimolar, which is very close to a physiological concentration of calcium in the extracellular space. And that means that when potassium in extracellular space goes up, it activates astrocytic specific sodium-potassium pump. And you can see it in the insert, and there are recordings of intracellular sodium. In neuron, when potassium goes up, nothing happens. In astrocytes, sodium goes down. Why? Because sodium-potassium pump is activated. And this is a major mechanism for potassium buffering in physiological conditions. In physiological conditions, you know, every action potential is associated with potassium efflux, and this potassium has to be taken off somewhere. And it is taken off into the astrocyte by sodium-potassium pump. And when neuronal activity ceases, this potassium goes back to neuron, and now it's released through inward rectifier potassium channels. So with potassium, there is a sort of a shuttle when potassium goes into astrocyte during neuronal activity and goes back to neuron at the moment neuronal activity stops. Another fundamental mechanism which is sodium dependent in astrocytes is a glutamate transporter. Now, astrocytes remove about 80% of glutamate, which is released during neuronal glutamatergic transmission. And glutamate, as you all know, it's not only a major neurotransmitter, but it's a natural neurotoxin, because if glutamate stays around neurons, it opens without desensitization NMDA receptors, which causes massive calcium influx, depolarization, secondary calcium influx, and neuronal death. So glutamate has to be removed, and it is removed mostly by astrocytes, which express two types of glutamate transporters, EAT1 and 2, with some regional differences, but these transporters are very similar. They are sodium-dependent transporters, so they take into the astrocyte 1 molecule of glutamate together with 3 molecules of sodium. Now, when glutamate comes to astrocyte, it is converted to glutamine. And another thing which is of fundamental importance, neurons cannot produce glutamate because they don't have certain pleiotropic enzymes which cause excess of alpha-ketoglutamate. So all glutamate in the brain is synthesized and after it is synthesized, again, it is converted to glutamine with an enzyme which is known as glutamine synthetase. And then it goes to neurons where it is now converted to glutamate and GABA. So without astrocyte, there is no neurotransmission in the brain. So sodium-potassium pump, how it works, it triggers massive sodium influx into astrocyte together with glutamate. And this sodium influx now has numerous feedback functions. First, it stimulates transporters of glutamine. So when neurons are active, they receive more glutamine for conversion to glutamate. Second, it stimulates sodium-calcium exchanger and triggers calcium signals in astrocytes. Third, it reverses GABA transporter, and astrocyte starts to release GABA. And that is more or less tonic inhibition, which is really important. So astrocytes are contributing to the major cycle of glutamatergic and GABAergic transmission through sodium-dependent glutamate transporter. And this is just to show you how big and how hugely capacious is this particular mechanism. So that is a Bergman glia, which is stimulated by glutamate and in conditions when glutamate receptors, of course, are blocked. So you see a current, and that is an inward sodium current generated by the transporter. And you see it saturates at 10 millimolar of glutamate. So there is a huge excess of glutamate transporters in astrocytes. The density is about 10,000 glutamate transporters per 1 square micron. And that could be compared only with density of sodium channels in the node of Ranvier. Our brain contains about 1.5 grams of dry weight of glutamate transporters. And these transporters are fundamental for the survival. Without these transporters, everything is dying. And that it happens, I mean, also in situ, and that's experiment from brain slice, and you can see how this transporter works and how it triggers this long-lasting sodium signal. So whenever there is neuronal activity, there is a massive sodium influx into astrocytes. And when sodium is changing in astrocytic cytosol, it controls all these homeostatic transporters. It controls pumps, it controls potassium channels and potassium transporters. It controls neurotransmitter homeostasis, all of them, adenosine, noradrenaline, serotonin, you name it, they're all sodium dependent. It also controls glutamine synthetase. It controls energy production through mitochondrial sodium-calcium exchanger. It controls pH because sodium is controlling proton exchanger and bicarbonate exchanger, and transport of different precursors, including, for example, L-serine, which you know, the D-serine is a coagonist of NMDA receptors, but it cannot be produced without astrocytes, because astrocytes produce L-serine, which they then send to neurons to generate D-serine and produce all these actions. So sodium signaling is of paramount importance for tailoring neuronal activity with astrocytic homeostatic response. Now we come to chloride, which is also a very important ion, and at the very beginning, as you remember, I mentioned that chloride concentration in astrocytes is very, very different compared to neurons. It's about 10 times higher. So in astrocytes, resting concentration of chloride could be as high as 30 to 60 millimolar. And there are a number of molecules which are producing chloride fluxes. And from those, of course, we have to mention channels such as chloride channels and bestrophin channels, which are calcium-activated channels. And we have to mention GABA receptors and glycine receptor, because astrocytes contain lots of GABA-A receptors, ionotropic chloride channels, and glycine receptors, especially in the spinal cord. And those are co-activated with neuronal receptors in the course of transmission. And what is really important when we start to look for chloride concentration in vivo, it turns out that it is brain state dependent. So sleep and wakefulness are associated with changes in intracellular chloride. So chloride goes down in a wake state and chloride goes up in the sleep. And this chloride is also highly dynamic, and when the animal is moving or when the animal is stimulated, that is associated with major chloride changes in astrocytes. Very dynamic, going up and down, state dependent, all these things, and what it all means. So this is an inhibitory synapse. An inhibitory synapse obviously has presynaptic terminal and postsynaptic compartment, and in the postsynapse we have GABA-A receptors. And when GABA is released from the presynaptic terminal, it opens GABA receptor in the postsynapse, and that triggers chloride influx into the neuron, which hyperpolarizes the neuron, and that is electrotonic inhibition. However, when it happens in physiological conditions in the cleft, this massive chloride influx into the neuron depletes chloride in the cleft, and that limits inhibition. But fortunately, nearby we have astrocytic leaflets, which are carrying the very same GABA-A receptors, and astrocytes have a very high concentration of chloride. So when GABA is released from neuron, it also opens GABA receptors in neighboring astrocytic process, and that triggers massive chloride efflux. Therefore, it enriches cleft with chloride and maintains inhibitory transmission. And that is indeed the case. So if we start to manipulate with chloride inside the astrocytes by specifically expressing either chloride channels or chloride pumps, which could be controlled optogenetically, you see that whenever we are interfering with intra-astrocytic chloride concentration, for example, increasing this concentration, that would affect neuronal activity. The more chloride is in neurons, the higher is the degree of inhibition. And vice versa, if we deplete astrocytes from chloride with optogenetic channels, then inhibition is suppressed. So transient fluctuations in cytosolic ions are the mechanism and substrate for astrocytic excitability. And this excitability is controlled by multiple ions. There are, of course, calcium ions, which do something which we don't know. There are sodium ions, which control astrocytic homeostatic response and tailor this homeostatic response to neuronal activity. And there are chloride signaling, which is central element in maintaining inhibition in the central nervous system. We also probably should probe for potassium because there are major potassium fluxes in astrocytes, but so far it's difficult to measure. And we also have to remember about bicarbonate and about protons, which are also mainly controlled by astrocytes and also can carry a signaling function. Now, Valerio, how much time do I have?
I don't know exactly, but I think that you have more 5 minutes more, 5-10 minutes.
Okay, so 5-7 minutes.
Yeah,
What I want to show you, I want to finish this with a little bit of experimental work. And I want to talk about ependymoglial cells, which you remember I mentioned where we were talking about classification of astrocytes, and show you how these ependymoglial cells are controlling an outflow of CSF from the brain. And ependymoglia was actually the very first glia discovered, and that was made by Purkinje and Gustave Gabriel Valentin. And here you can see the very first image of glial cells, and these are ciliated ependymoglial cells. You see the cilia, which are protruding into the ventricles, and those cells are very peculiar. To a high degree, they have a signaling physiology very similar to astrocytes. They are connected with Glia junctions, they form a syncytium, they have metabotropic receptors, and they have calcium signals and calcium waves. But on top of that, these cells are also receiving synaptic innervation. So they receive glutamatergic synapses, normal GABA, ionotropic glutamatergic synapses, and they receive lots of contacts from serotonergic neurons. And here you can see that. So these are contacts, nervous ending, which are sitting around the central canal of the spinal cord, and which are sitting precisely at ependymoglial cells. And at EM level, we can find synapses which are ending at ependymoglia. And ependymoglial cells also express number of receptors to monoamines, and in particular here you can see serotonin receptor of HT2B subtype, and you see that ependymoglia are absolutely full of these receptors. And what happened when you stimulate those catecholaminergic or serotonergic receptors? This glial cell, this is culture now, they shrink. So what happens they change the volume. Why do they change the volume? Because activation of this metabotropic receptor triggers calcium signaling in ependymoglial cells, and this increased calcium triggers polymerization of F-actin. So these cells are shrinking. So they're actively responding to innervation of serotonergic fibers, which are coming from the brain throughout of the spinal cord. Now, CSF, of course, you know about it. CSF is produced by choroid plexi, and per day we are producing about 500 to 600 milliliters of CSF, whereas the whole volume of ventricular spinal cord system is about 120, 150 milliliters. So it means that it's a highly dynamic system. A new CSF is produced, and then all CSF has to go away. And there are many different outflow pathways through lymphatic system, for example, through arachnoid granulations to lymphatic vessels, and so on and so forth. But part of it goes to the spinal cord through the central canal. And we ask ourselves the question, can this particular CSF coming to the spinal cord reach peripheral organs through the spinal nerves? So that was an idea which was born about 5 years ago, and it took many years to go through. And, oh, the image has disappeared. Wow. So more or less, to make the whole story short, what we did, we injected a fluorescent tracer into the cisterna magna. And about 4, 5, 6 hours later, this tracer was identified in the liver, in the pancreas, and in the skeletal muscle. And then we did a crucial experiment in this situation, because how that can come to the liver? It can come with a blood flow. So we ligated the vena cava, and that reduced the amount of tracer in the liver by about 30%. But when we ligated all spinal nerves, that reduced the amount of tracer by 70%. And that somehow shows that CSF with a tracer can migrate from the central canal, from the spinal cord, from the CNS to peripheral organs. And how it does it? So first it goes to the central canal. Then from the central canal, what's going on with all these images? They disappear. It goes to parenchyma of the spinal cord. I'm very sorry, I don't know what it is all about. And we can then image it. So this is imaging of dorsal roots of spinal nerves. And these nerves, the axons are green because they are expressing UFP under control of neuronal promoter. And tracer is red. And here, as you can see, when we image this particular cell, we can image the transport of tracer through spinal nerves. And this tracer, sorry, that's really bad. I don't know why. More or less, this tracer ends up in the liver. And that is regulated by serotonergic receptors and by stimulation of serotonergic neurons, which are sitting in the raphe nucleus. So that is just showing you the final scheme, how CSF flow to the periphery can be controlled by ependymoglial cells. When ependymoglial cells are shrinking, they open water channels, and more CSF is redistributed to the peripheral organs. And that, of course, is of paramount importance of both physiology and pathophysiology, because CSF contains very many signal-competent molecules. It contains hormones, it contains metabolites, it can contain pathological molecules such as, for example, beta amyloid. And in this way, it produces a new pathway for body-wide volume transmission, in which things which happens in the brain could be sensed by peripheral organs. And so with that, I'm going to finish my lecture, and thank you very much for your attention. Right, how do I stop it?
Thank you very much, Alex, for your amazing presentation that cover all the different glial cells, not only glial cells, because in the last part you mentioned also these very super new data about epidemioglial cells.
I'm terribly sorry for, I don't know what happened to that, because obviously when I tested it half an hour before the lecture, everything was fine.
Never mind, maybe it's linked to, it's due to Meet or this kind of connection that is, maybe you are much more used to Zoom, but
usually I use Zoom, I don't know how it works, and that is the first time, so but I have to check. Anyway,
never mind. I think that Nermeen want to
Okay.
yeah, want to
Yeah.
organize
Okay.
this discussion, or whether someone.
I'm okay with it. If you want to go with the questions, or Professor Alex, do we have some a couple of minutes for
Oh
questions?
yes. I'm not in a hurry whatsoever. So
Okay, thanks a lot.
it's
Thanks a
more or
lot.
less it's Friday, December, so it's nowhere to go.
Yeah, thanks a lot for this engaging webinar, and I think it inspires a lot of questions already. So here's a question. Maren wanted to ask about calcium imaging in astrocytes, which is a common method. And in neurons, we use it as a proxy for activity because of its involvement in the release of vesicles in the action potential process. But in astrocytes, can you please elaborate on the meaning of calcium in astrocytes as a proxy for activation and calcium imaging? Is it needed for imaging or the release of the other neurotransmitters? And is the internal release of calcium from the endoplasmic reticulum in astrocytes correlated with something we can define as activity? Thanks.
Well, that's a wonderful question, and indeed it all began, and I was very, very lucky because I joined the glial community in 1989, and there were the very first setups for measuring calcium and everything, and that was highly exciting. You take an astrocyte, you put it under the microscope, you inject, you put something on top, and whatever you put on top, you have calcium rate. So in a sense, we tried, I don't know, and people around the world, they tried all sorts of neurotransmitters, all sorts of hormones, everything. And everything inevitably triggered calcium signal.
Yeah.
And that more or less was the basis for the whole idea that this is a universal mechanism for excitation. And yes, even if you go in the slices and even if you go in vitro, the neuronal activity and activity of the brain and stimulation of different sensory inputs or motor function, they do trigger calcium signals in astrocytes. So yes, it's a proxy of astrocyte activation, which is also very important because activation is physiology. When we go to pathophysiology is reactivity, so we should not mix up those words. The question is, what is a specific function? So in neuron, everything is simple. Calcium, exocytotic release, neurotransmission. What astrocytes are doing? And that is a major question. So we still are a little bit in the dark of how it is encoded and decoded in the functional properties. Of course, astrocytes also have secretion, yeah?
Yeah.
And its secretion is much more complicated than just release of ATP. So astrocytes release hundreds of different molecules. And this release could be vesicular, or could be through transporters, or could be through diffusion. And those processes, of course, could be calcium dependent. So the question is really how to pinpoint specific functions which are calcium dependent. And that is a major challenge, and very many groups are working on it.
Okay, thanks a lot. Gabrielle is also reflecting on Maron's question, and she's asking about the use of chemogenetics in astrocytes, because there's increasing use of such a stimulation in astrocytes, and she is wondering, or Gabrielle is wondering, what your thoughts are on this, whether it actually taps into a physiological process, and what conclusions we can draw from such manipulation.
Yep. First of all, of course, it's a major revolution because now we can really image astrocytes in the brain, and we can see reporters and we can see fluctuations of different ions. So for the time being, we have calcium, we have chloride, for example, we have sodium. We can look for fluctuations of second messengers and so on and so forth. So that is already a very serious thing because now we can specifically measure them in astrocytes. There is, however, the second layer, and that is optogenetic or chemogenetic manipulation. So, for example, when we express new channels and new pumps and new transporters, whatever it is, and then we manipulate with them with whatever things. And then we have a response. And quite often there is even a behavioral response. But here comes the question, the fact that optogenetic stimulation of astrocytes will trigger behavior response does not necessarily mean that this response is existing in an animal, because in a sense, we introduce a new system which may produce a new response, which everything works, but it's not biological, it's sort of irrelevant in a sense. It's still extremely interesting. And that also even more important when we go to pathology and pathophysiology, in which we have to make disease models. And what I am afraid we do, we create disease models, but this is not a model of human disease. We are just making a new disease of mouse with obvious limitations.
Yeah. Great answer. Yeah. I have a question about the chloride dynamics, and in pathological disorders, does disrupted chloride regulation can affect the glutamatergic mediated excitability or neurotoxicity before it affects the neuron chloride homeostasis or something like this?
It's a very, very good question. Now, I cannot give you the answer because so far, well, there are only 2 papers measuring chloride in astrocytes in general. Yeah. But of course, we are working on it. What is called ionostasis, so let's say homeostasis of all ions, are absolutely intrinsically coupled. So whenever one ion changes, everything else changes too. So it's absolutely conceivable that in pathology we will be dealing with alterations of chloride homeostasis.
Yeah.
And in particular, it's important, for example, for traumatic things when we have a penumbra around stroke. Yeah. And in this penumbra, all the ions are messed up completely.
Yeah.
But how they messed up, we don't know.
Yeah.
We know that they are messed up.
Yeah.
But how and what is the primary, what is secondary, and how they are all interacting, and how that affects inhibitory and excitatory transmission and the balance, this is, of course, a major challenge. So, but I am absolutely convinced that, I mean, what we need to do, we have to study probably all ions simultaneously in the same context.
Yeah. Yeah, and this brings us to the neurodegenerative disorders with the astrocyte. Do they lose their supportive activity first, or do they change their dynamics into something like toxic or acquire toxic properties so they can?
Brilliant question, and I'm afraid, Valerio, probably we will need to do another lecture about pathophysiology of astrocytes.
Yeah.
So, it
Please.
is really an extremely interesting discussion and controversy which lasts probably 20-25 years. And that is the question: can neurons or can any cell in the brain become toxic? So what does it mean, toxic? It means that it synthesizes a toxin de novo. Now, for that, we don't have any credible evidence.
Yeah.
What we have lots of evidence that loss of homeostatic function triggers neuronal death. The simplest example is amyotrophic lateral sclerosis. Why neurons are dying? Because in astrocytes, expression of glutamate transporter is decreased by 90%. Done. In this context, the most neurotoxic cell in the brain is neuron, because it releases kilograms of glutamate. Yeah? But we don't call it neurotoxic. At the same time, we call microglia neurotoxic, astrocyte neurotoxic, and it comes to a very distorted image that all these cells are sort of dormant killers. They are sitting in the brain and just waiting. Something happens, they immediately go out and kill everybody. Nothing could be more wrong, because their main function is to protect.
Yes.
And if you think about it, during our life, let's say 70, 80 years, we experience multiple pathologies. There are probably thousands of nano-strokes or whatever it is, and infections and cytokines, and we don't care. Only at the very end, when we die, then something goes wrong. So in a sense, as long as protective systems are working, everything is fine. Unfortunately, our studies of pathology are highly biased towards insufficiency, because we look at the point when they did not work, and that is death. And we never look at the point when they did work and nothing happened. And that, of course, creates probably quite. I mean, it's very difficult then to interpret and to do all these things. Well, again, for example, in Alzheimer's disease, one of the pathways of degrading beta amyloid is associated with urea cycle. An urea cycle is astrocyte specific. And when there is a beta amyloid load, this urea cycle starts to work more, and it does whatever things and takes ammonium and takes beta. But as a side effect, there is more reactive oxygen species.
Yes.
However, if astrocyte would be fine, it will respond with increase in glutathione production, because that's what astrocyte does.
Yes.
However, it's probably stressed, and it doesn't. And in this situation, this reactive oxygen species obviously become toxic. So it's in all cases, we have probably a protective mechanism which runs as far as it can. And if this protection cannot work anymore, we have accumulation of potentially toxic molecules.
Yeah, yeah. Thanks a lot. And another question from Giuseppe: to what extent is astrocytic calcium signaling necessary and sufficient for the release of gliotransmitters in vivo? And should yeah, and should and should calcium astrocyte be interpreted as instructive signal or mainly as a marker of homeostasis?
It's impossible. I mean, the last part. It's impossible to say what is instructive or is not instructive, because astrocyte can actively affect neuronal transmission, as well as neuron can actively affect. So all the cells there and they do actively affect each other. So I think that it's impossible really to distinguish between homeostatic part and signaling part. And that applies to every cell. It also applies to the neuron, which, for example, controls functional hyperemia. Yeah? So it's also instructive and at the same time homeostatic. So I think we have to somehow change our mind, and it's very difficult. We have to look into all that in its entity. It's not that this cell does that. All of them are doing different things, and they're dynamically regulated. When it comes to a concept of glia transmission, it's again, it's highly controversial. Well, glia transmission exists, there's no doubt about it. So glial cells do secrete many molecules which are signaling molecules. Somehow, most of the attention is now paid to glutamate, to ATP, and to the LD serine. Yeah? When it comes to glutamate, there is absolutely not a single experiment which shows that astrocyte can release glutamate, which will trigger action potential in neuron. That's all. So even if this glutamate is released, it is not for making signaling in the neuronal network. It may do some local stuff, no problem. At the same time, we are forgetting about other many molecules which astrocytes release, like thrombospondins, like heavins, like S100A6, whatever it is, there are many, which are signaling molecules. So I think that we need to look into glia transmission again in a much wider scale and identify transmitters which are specific to astrocytes. Glutamate is actually neurotransmitter, and it does not matter whether it's released by astrocyte or neuron. It remains neurotransmitter. But thrombospongin is a classical gliotransmitter because it is not released by neurons. So I think that the whole issue here needs rethinking and remodeling, but yeah, very important.
Yeah. Okay. One last question, I think, about the ganglion sensitivity, and does it is it regularly or dynamically regulated, or fixed, or or developed with sleeping, learning, with aging? Is it affected? Because I think it's very exciting to know more about. Yeah, and,
absolutely. I think.
And whether the brain organoids can give us more reflection, more like imitating the astroglial microenvironment more than the animal models, which are with this lecture, it's totally different from the human physiology.
Precisely.
Yeah.
Well, Obviously, so there as for neuronal things, there are some anatomical things. So there are probably some pre-developed pathways for syncytia, and there are functional things, and they are probably plastic.
Yeah.
And of course, you can manipulate with gap junctions in a very wide range. So they are phosphorylated, they are subjects for different regulations, and we know that we can close gap junctions, we can open gap junctions, and yes, of course, it's plastic. So during lifetime, obviously, it is changing.
Yeah.
And in my view, it's like two different systems. One is neuronal connectome, and one is panglial syncytium. And again, they work together, and depending on what's going on, they remodel themselves together. Can we image it in, well, so far we don't have cellular imaging, of course, in vivo imaging. So that's the main question. And yes, I think that we have to humanize research. We have to somehow more human-centric research, but it's obviously very, very difficult.
Alex, I cannot thank you enough for this astounding webinar and discussions following this amazing lecture. Thank you again. Thank you for sharing your expertise and your time with us. And we are very grateful for this webinar indeed.
Thank you very much. It was fun and joy, and I wish you all Merry Christmas, Happy New Year, and all the best in your lives. Thank you very much.